Hello, my name is Steffen Hansen. Thank you for joining us today as we discuss plant-based concentration using membrane technology. A little bit about myself. I am subject matter expert in membrane filtration and microparticulation at SPX Flow. I've been with the company since 2019. I have a background in process technology and food technology. My responsibilities within SPX Flow include dimensioning of commercial scale plants, technical sales support, troubleshooting on-site and remotely, doing pilot trials either internally or with customers, as well as developing new products for our portfolio. It is my goal that everyone will be able to take away something new about plant-based concentration and learn how our APV membrane filtration solutions may help you in your role or application. A few housekeeping notes before we get started. As an audience member, you will not have audio or video capability. Please use the Q&A feature to submit any questions throughout the presentation, and I will answer as many as I can at the end. If we do not have time to answer them all, my contact information will be provided during the Q&A session during the last 10 minutes or so of this presentation. So let's get started with a discussion on sustainable plant-based processing membrane filtration. First of all, I'd like to remind you of SPX Flow overall purpose, which is to improve the world through innovative and sustainable solutions. The agenda for the next 20 so minutes will be the following. First, I'll introduce the oat beverage process. Then I will talk about some of the challenges that we see in plant-based processing. Next, I'll go to membrane filtration, explain you briefly what the technology is about, and then I'll talk about how we can use it in plant-based applications. Next up, I'll summarize briefly what we've learned so far from the plant-based trials we've done with membrane systems. And finally, I'll show you how you can design and develop your process alongside us. So here's a flow diagram of the overall beverage process, for example, oat beverage, where we start with grains and grits as the raw material. We go through enzymatic treatment, decantation. If we have to mix in ingredients, we'll use a mixer. Next up, we'll do UHT treatment typically, and then homogenization before the product is aseptically packaged and sold. Now, you may notice there's not a filtration system in this slide yet. That's because we have not sold any commercial scale membrane plants for plant-based applications. But if we had to include a membrane plant, it would probably be after the decanter, where we could imagine to increase the protein content, for example, by membrane filtration. Here are three key challenges that we see in the plant-based processing. First of all, every crop is different in terms of composition, fibers, solubility, etc. So that's a challenge to see that every time we talk about a new crop, we need to consider different pre-treatments. I'll get back to this later. Secondly, there's a huge variation even within the same crop. So you have different cultivars, you have different extraction history, different upstream heating history, just to mention a few of the variations that we see. Thirdly and lastly, the cleanability and processability can be challenging due to the presence of dietary fibers, starch, protein agglomerates, etc. So, introducing APV membrane filtration. Membrane filtration is a way to increase the nutritional value of your product, leading to cleaner label, for example. It can also enable increasing yields, waste reduction, and extend shelf life, offering a great flexibility. So, what is membrane filtration? If we put it very simply, it's a pressure-driven separation of components based on their molecular size. We use cross-flow filtration across the membrane surface as a way to optimize flux and by preventing fouling. So if you see this figure in the top, you have to consider three streams in the membrane filtration system. Of course, you have a feed stream. And then the membrane, depending on the pore size that you choose, will cause a separation of the different components in the feed, depending on their size and, of course, the pore size. The components that are retained, that means they are bigger than the pore size, we call it retentate. And the components that permeate the membrane because they have a size that is smaller than the pore size, we will call the permeate. And traditionally, we have 4 categories of membrane filtration, depending on the pore size. And those are microfiltration, ultrafiltration, nanofiltration, and finally, reverse osmosis. What's the difference between these 4 pore sizes? Well, as you might have guessed, microfiltration offers the largest pore size. That means we can retain only suspended solids and bacteria. Ultrafiltration is a little tighter. That means we are retaining large molecules. Nanofiltration is the second tightest membrane, so to speak. So you'll also retain small molecules. And reverse osmosis is technically a membrane that is impermeable to anything but water. Some more generalities about membrane filtration. We have different configuration of the membrane filtration inserts. So we can consider, for example, spiral wound, that's the top right corner, tubular ceramic, that's the top left corner, hollow fiber, bottom left, and finally plate and frame. And each of these configurations have their own, you can say, opportunities and where they are very strong, but they also have their downsides. That can both be economics and in terms of the viscosity they can handle. I'll get back to this later. So what opportunities do we see by applying membrane filtration to plant-based products? We actually think we can achieve some of the same wins as we have already seen for dairy. So we know that membrane filtration is a wet extraction process. It can give you better protein yield and sustainability compared to a conventional method of isolating proteins. You can preserve the nativity of the proteins, eliminate non-protein anti-nutritional factors from soy beverage, for example. You'll remove the water-soluble proteins and recover them. You can produce protein isolates. That means at purity of more than 90% protein. You can valorize oligosaccharides, which is a form of sugar. Membrane filtration is often seen as a more sustainable way than, for example, chemical precipitation of proteins. Finally, we also think that we could maybe reduce the bioburden and spores to give an extended shelf life of the final product. So to summarize it in 3 keywords, we think we can improve, give a higher nutritional value. We think we can give a better yield, and we also believe that we can offer a sustainable solution. If we consider a membrane filtration plant in a plant protein production line, it could look something like this. Again, I'm stressing this is not an installed base, but this is how we think we might be able to include it in a process line. So after the hydrolysis tank, you have a decantation, you have the membrane system, then you'll optionally have a separator to further purify. Then you'll go through several tanks and an evaporator, and then spray drying the protein isolate into powder finally. Now I'll talk a little bit more about the practical lessons learned that we have from many of the plant-based trials we've conducted within SPX Flow. So one of the key findings we found was that plant-based is not a one-size-fits-all. We were probably initially hoping that if we, let's say, did 10 trials on oat beverage, then we would be able to apply that knowledge to, for example, soy or pea beverage, but it turns out not to be true. First of all, let's look at the different families that the plant-based are split into. We have the legume-based, the cereal-based, the ancient cereal-based, the nut-based, and the seed-based. All of these have different properties and compositions, as I'll show you in a bit. The raw materials or the crops that are shown in bold are the ones that we have hands-on experience with. So if you're interested in any of these applications, please reach out to me after this webinar. But to give you an example of the variability for these raw materials, we can look at a nut crop such as almond, which is very oily and has a lot of protein. In contrast to that, oat is dominated by carbohydrates, so that would be starch. Soy is a good mixture of a lot of protein, a lot of oil, and some carbohydrates. Whereas yellow pea is very rich in proteins and to a lesser extent carbohydrates. So this shows you the variation between different types of raw materials. The next finding we did was that even within the same crop, we saw huge variability. So this is an example where we did a market survey. We bought 14 commercial oat beverage samples from the European market and looked at it in various ways to find out what is exactly the variation. So looking at the particle size for these 14 samples, for example, we would probably have expected to see, at least within oat, that they would be more or less on top of each other, but no, we found very big variation from some containing small particles and some containing big and some containing a good mixture of everything. The same picture showed under the microscope, where these are micrographs of 3 different samples. The left one, we see something that looks like undissolved gelatin gum or chicory fiber, which is in the ingredient list. The one in the middle seems to be oat fibers, and the one to the right seems to be unhydrolyzed starch granules. So again, this shows you that even within oat, we have many, many different particles and to take into account. This means that the correct pre-treatment is really key before we do any membrane filtration, because the main obstacle of using membrane filtration is the presence of large particles, that means 100 micron or more, such as fibers, starch, oil bodies, or agglomerates of proteins. And you can see again, just to show you some examples of what can be found in the different raw materials. In the P, we have what seems to be starch granules, or at least some cellulose structure. The quinoa has another type of structure in it. Hemp is very dominated by oil bodies, and oats has both fibers and oil and starch, of course. So we set out on a set of experiments to determine what exactly is needed in terms of pre-treatment. Would it be physical methods such as decantation, clarification, and skimming? Could it be homogenization to break down some of the structures? Or could it be an enzymatic treatment to maybe hydrolyze the fibers? What it showed was that the pre-treatment was, of course, very essential, but also the choice of membrane configuration. As I showed you before, there are 4 different types of membrane filters. One of them is spiral wound, and it seems from our findings that this membrane time is not compatible with all types of plant-based products. It is often the spiral wound is often the go-to membrane type because of its low cost and its high capacity compared to the volume that it contains. But in this case, we've seen that after the CIP of this trial with a plant-based beverage, we still see a soiled membrane surface. The pollutant seems to be of a fiber origin when we look in the microscope, but the conclusion from this trial was that this specific plant-based beverage, even though it was pre-treated, would not be compatible with spiral wound membranes. In another set of trials, we wanted to make a kind of proof of concept and find the most optimum combination of membrane configuration and pore size to be able to concentrate the proteins in a plant-based beverage. So the first leg of this trial was to screen for the most suitable or most economical membrane type and pore size. We tested ceramic membranes, hollow fiber membranes, and spiral wound membranes, both in microfiltration, ultrafiltration, and RO. Next up, when we found the most suitable membrane type from the pre-screening, we did a runtime evaluation to see how long could we possibly process these products before we had to do CIP. And we also arrived at a preliminary design basis for a commercial plant. These are some photos from our pilot facilities here in Silkeborg, Denmark. So next up, I'll summarize very roughly what we found. For example, for oat beverage, we arrived at a membrane configuration and pore size that allowed us to concentrate the proteins 10 times alongside the fat and, of course, some residual starch. So what you see in this picture is that the oat feed becomes much more viscous, much more opaque, and more, you can say, the brown color is really amplified in this case. Now, the permeate, on the other hand, is totally clear, and it's very sweet, so that means it contains primarily sugars. And of course, if the retentate is the main product, we also have to look at the byproduct, in this case the permeate, and how to valorize that by some means. But it's important to say that we now have a cleanable solution with the right membrane configuration to be able to provide this this solution for a oat beverage. If we look into soy beverage, we did other trials where we arrived at the right configuration. We were able to concentrate the proteins in the soy beverage about 2 times. So we go from around 5% protein to 10% protein. Again, the retentate changes appearance and viscosity compared to the feed. And the permeate is very clear again and contains primarily sugars. So a cleanable and viable solution for soy beverage concentration. The final example I'll show you here is from yellow pea protein isolate, where we were able to increase the purity of protein. So we start out something like 60% protein to total solids ratio. And by using the right membrane type and pore size, we were able to achieve up to 80% purity. That's without diafiltration water, so it's possible to go even further. Again, retentate looks different compared to the feed. We have retained all the proteins and some other stuff, whereas the permeate contains primarily sugars again and is completely clear. So a viable and cleanable solution for yellow pea protein isolate production. I'll now tell you a little about our testing capabilities, because with us you are able to design, develop, and perfect your manufacturing process. So first of all, we have innovation centers around the world. 3 of them have membrane systems and are able to support plant-based trials. We have an innovation center here in Silkeborg, Denmark. We have one in Membrane in France. And finally, we have one in Shidu in the Shanghai area of China. Secondly, you can also rent one of our membrane pilot plants to get them on site. That's a chance to perfect your recipe before you upscale to commercial production. And the benefit of renting the pilot plant is that you have access to it over a longer time, and you can also use raw materials in bigger quantities than if you had to ship it to one of our innovation centers. Finally, if you decide to purchase a commercial scale membrane plant, we offer service agreements, which is a way to protect the life and efficiency of your investment. So I want to end off this presentation by saying that in conclusion, we are SPX Flow are very well prepared for plant-based applications within membrane filtration. We are ready for a commercial partnership, and we welcome you to contact us with your ideas for future applications. So all in all, by looking at the right picture, it's time to take your plant-based products to a higher level. I want to turn your attention to our other plant-based processing webinars. So this is a list. Some of them are already available and some will be published later. This ends today's presentation, and I will now begin our questions and answer session. Just a quick note, after we complete the webinar, all participants will receive a copy of this presentation and ability to submit a private question to me and my team. We welcome your questions and commentary on your plant-based projects. Okay, I'm ready to answer some of the questions. Hopefully I can answer them all. I see here there's one question about slide 20 and 21. The question goes, if I apply membrane filtration to soy and oat products, my permeate will consist primarily of sugars. Does that mean that my retentate does not contain sugar? In fact, it doesn't. You can, as a rule of thumb, you can expect that the retentate will have the same amount of sugars that you have in the feed. However, the composition will be changed because you have then concentrated other components such as proteins and fat. So the answer is no, you'll have the same concentration of sugars as you have in the feed. Another question goes, are special CIP detergents needed to clean a membrane plant? And I guess by special CIP detergents, you mean something that's not simply sodium hydroxide or hydrogen or nitric acid that you use for the other processing equipment. The answer is yes, because the membrane system contains, we rely on these membrane elements that are typically made of plastic or polymer material that is not tolerant to high pH and high temperature, or low pH and high temperature. So we actually need specialized CIP chemicals to be able to clean them properly, and that increases the cost of a membrane plant, the operating cost of a membrane plant. Are RO systems more efficient than evaporators for removing the water? That's a good question. It actually depends. If you have an existing evaporator with MVR technology, that means very efficient recovery of the heating energy, then the evaporator will be more efficient than RO. However, if you're looking into buying, if you are looking into concentrating, for example, sweet whey or whatever plant-based beverage 2 times, the RO system may be competitive because it takes up a smaller footprint. It's cheaper to buy, and if the you can say if the in terms of utilities, if the energy is cheaper than the steam, then the RO system will be a more cheap solution, at least in initial concentration stages. And at some point, the evaporator will become much more profitable because the flux of the RO plant will be so low. So the answer is both yes and no. Next question goes: what is the main operation expenditure associated with a membrane filtration system? I would say the main expenditure, as with other systems, is of course the utilities. But when we talk membrane filtration systems, we also have to consider a relatively high cost for the CIP detergents. So they are probably something like in the order of 25 to 30% of the total operation expenditure. And then you also have to consider the lifetime of the membrane elements, because they are not, since they're not made of steel, they are a polymer material, so they have to be replaced at a certain interval, and that actually also contributes somewhat to the OPEX of a membrane system. Okay. Next question goes: will bacteria removal by microfiltration produce a 100% sterile permeate? And the quick answer to that is no. A microfiltration membrane is not a sterile filter, but it has a very high log reduction. So it can be anything from log 3 to log 4 or even 5. So it means you'll significantly decimate your bacteria count, but you cannot ensure a 100% sterile permeate. Then you need typically a downstream pasteurization to achieve the final log reduction. Yeah. Okay, I don't see any more questions ticking in. I'll just give the audience 10 or 15 seconds to submit a question. So that is all the questions I see for now. Thank you very much for joining and for your engaging questions. We appreciate your active participation and look forward to connecting with you again soon. Thank you for joining, and goodbye.